actually the wrong one. I have to go back.
Well, to start with, I would like to welcome everybody, also the audience, to join us today. And hereby I open officially this academic ceremony in which Ms. Anja Schöittler will defend her academic thesis entitled The Role of Complement and its Relevance as a Therapeutic Target in Myasthenia Gravis. And I would like to ask you to present a summary of your studies and the conclusions of your thesis in the next 15 minutes. And I'd like to give you the word.
Thank you. Dear Pro-Rector, dear members of the Corona, dear colleagues, families and friends. In the upcoming 15 minutes, I would like to show you the highlights of my PhD research written down in my thesis entitled The Role of Complement and its Relevance as Therapeutic Target in ACHR-MYASTHENIA Gravis. The immune system defends the body from intruders like viruses or bacteria. For this, it developed several different strategies. One of these strategies is the production of antibodies that find and eliminate specific pathogens. Even though antibodies work already very efficient, they do not fight alone. To support the antibodies, proteins called the complement system come for help. A leading role within the complement system has the protein C3. C3 gives the command to perforate the pathogen with the so-called membrane attack complex, here represented by the flash. The membrane attack complex is a protein that makes little holes into the pathogen membrane. The effect is easy but efficient. The attacked pathogen is destroyed. However, the antibodies can be programmed wrongly, and instead of fighting against pathogens, they fight the body's own cells. The immune system produces autoantibodies against itself. This is called autoimmunity. In my PhD projects, I focus on the autoimmune disease acetylcholine receptor myasthenia gravis. In the disease myasthenia gravis, muscles cannot fully receive the information from the nerve, leading to severe muscle weakness. But to explain what actually happens, I first would like to explain the situation in a healthy muscle. The nerve meets the muscle in an area called neuromuscular junction. At this place, the nerve can send information to the muscle. However, there's a little gap between the nerve and the muscle, which the information needs to cross. For this, the information is packed into little packages, the neurotransmitter acetylcholine. The acetylcholine is released from the nerve and travels through the gap towards the muscle. At the muscle, the acetylcholine binds to the acetylcholine receptor that processes the signal and turns it into muscle activity, for example, an arm movement. The signal transmission between nerve and muscle could be compared to sending a letter. The sender writes a letter, sends it by mail to his friend, and at the friend's house, the letter is delivered into the mailbox, and the information can be processed. Coming back to myasthenia gravis now, the transmission of the information is impaired by the autoantibodies that bind and block the acetylcholine receptors. The acetylcholine cannot deliver the information. Besides the blocking of the receptors, the autoantibodies activate the complement system, which I explained earlier. The complement system now perforates the muscle membrane, removes the receptors, and impacts the muscle function. Due to these effects, myasthenic patients experience a muscle weakness. Speaking in the letter analogy, in myasthenia gravis, the letterbox is stolen. The mailman cannot deliver the letter. It will return to the sender and not transmit any information. Current treatment methods focus on immunosuppression. The aim is to reduce the amount of autoantibodies that could initiate the myasthenic muscle weakness. However, it also reduces those antibodies that protect us from infections, and with this, the patient is of higher risk to get ill. Therefore, improvement and especially the development of treatments that target specifically the disease mechanisms are needed. We followed up on this path, and in collaboration with Apellis Pharmaceuticals, we tested a compound that inhibited the expression of the central complement protein C3. We hypothesized that inhibition of C3 would prevent the formation of the membrane attack complex, preserve the structures and receptors in the neuromuscular junction, and with this, C3 inhibition would preserve the muscle strength in myasthenic patients. To test this hypothesis, my research was divided into different projects. Firstly, we tested the effect of C3 inhibition in the acute rat model as proof as concept. Afterwards, we tested different time points of treatment in the advanced chronic rat model. Additionally, we characterized and compared the myasthenic rat models to assess limitations and strengths. And lastly, apart from the complement-related research, we aimed to refine the chronic myasthenic mouse model, which is also very commonly used in myasthenic research. However, due to the limited time today, I unfortunately will not go into more detail of this last project. In my first study, we used the acute disease model to test our hypothesis. In this model, the animals develop weakness very soon. Within 48 hours, they get muscle weakness. We treated the rats for 2 weeks with the treatment to decrease and eliminate C3 from the circulation. Then, after 2 weeks, we injected the disease and analyzed, among others, the C3 levels in blood, the strength, and the muscle-damaging membrane attack complex. With the blood analysis, we could confirm that the treatment successfully depleted C3 from the circulation, meaning any potential symptoms in these rats should not result from complement-related damage. Analyzing the strengths, we saw very impressive results. While diseased animals were weak, diseased rats treated with the C3 inhibitor, here the green ones, remained strong, similar strong as the healthy control rats. And this clearly supported our hypothesis that C3 inhibition would preserve muscle strength. Let's now come to the presence of the membrane attack complex. We made the structures of the neuromuscular junction visible with 2 different markers. In red, we stained the acetylcholine receptors. And in the lower row, in green, we stained the membrane attack complex. It is important to mention here that the red and the green marker are exactly in the same location of the muscle. As you can see in the healthy rats here, no membrane attack complex is found. The lower image is basically black. In contrast, the diseased rats presented intense green stain, meaning there is membrane attack complex, and this was expected as these rats did not receive any treatment. But now it becomes interesting. In those rats that received the C3 treatment, we also found the marker. We also found the membrane attack complex in the membrane, at least in some locations of the muscle. This was very surprising because we successfully removed C3 from the circulation and expected therefore also that the follow-up, the downstream membrane attack complex, would also be removed. Nevertheless, we moved forward, and with the same hypothesis, we tested 2 different time points of treatment now in the more advanced chronic model. In one group, we tested the preventive treatment, where the rats received the treatment before disease start, to investigate the role of complement during the development of the disease. In the other group, we applied classic treatment, meaning the rats got treated only after disease onset. This showed us insights into the long-term effects of complement in myasthenia. The classic treatment, of course, is much more comparable to the situations in patients which also first get the disease and then get the treatment. Also in the chronic model, we confirmed that the C3 was successfully eliminated from the blood in both the preventive and the classic approach. And this clearly reduced also the severity of the disease. It is to mention that usually some animals in this model develop so severe symptoms that they are taken out of the experiment before the official end of the experiment. From the diseased untreated rats, around 30% developed so severe symptoms and were taken out. However, from both treated groups, the preventive and the classic, only 5% of animals dropped out. This indicates that in one way or the other, the treatment was effective to protect the animals from myasthenia. Quite euphoric of these results, of course, we analyzed the presence of the membrane attack complex. At least the healthy controls did not have any membrane attack complex, here in green in the lower row again. Unfortunately, we figured out that all the diseased animals expressed the attack complex. The untreated, the preventive treated, and also the classic treated, which was quite a surprise since C3 levels were so low and the disease severity was so promising. Even more surprising then was the result of the strength analysis. Since we had confirmed that the disease was properly developed, we expected also to observe chronic muscle weakness in the untreated diseased animals. However, in none of the groups we observed this weakness. All remained their strengths in terms of the experiment. Summarizing the results of both the acute and the chronic study, we can state that the tested C3 inhibitor is effective to eliminate C3 from the circulation, that C3 inhibition reduces the severity of myasthenic symptoms, at least if administered before disease start, and that the used C3 inhibitor is not effective to protect the animals from the membrane attack complex. If you remember back that the C3 gives a command to produce a membrane attack complex, we can conclude that there must be either an additional source of complement or an additional route of complement activation than the one we inhibited with our treatment. If those additional sources or routes in the chronic and the acute model are the same, this is another story. Initiated by the question why we did not observe chronic muscle weakness in the chronic model, we compared and characterized the acute with the chronic rat model. In both models, the membrane attack complex was present. With this, the muscle damage has happened and the receptor loss has happened, but symptoms only occurred in the acute model. Let me use the ladder analogy again to explain it. In both the acute and the chronic disease model, the muscle cell is damaged. The ladder box was stolen. However, in case of the long chronic model, we could prove that the nerve increased the production and the release of the acetylcholine. The ladder was basically copied several times. And the more ladders are sent, the higher is the chance that one of them would be delivered to another receptor, to a neighbor, and the information could be delivered anyway. This leads me to the general summary and conclusions of my presentation, which I would like to do in reverse order. We presented the importance of modal characterization and explained the preservation of strength in the chronic model. We are convinced that the inhibition of complement has beneficial effects to decrease the severity of myasthenia gravis, and in particular, C3 inhibition is a promising strategy as treatment in acute phases of myasthenia. However, for higher treatment efficacy, we suggest to investigate and target additional activation routes and sources of complement as well. And last but not least, in the beginning of my presentation, I mentioned the importance to find alternative and more disease-specific treatments for myasthenia gravis. And the complement system is a supportive part of the immune system, but a main driver in myasthenia gravis. We think that the treatment with complement inhibitors could preserve the protective antibodies while reducing the muscle weakness in patients at the same time. With that, I thank you for your attention and would like to give the word back to the Pro-Rector.
Thank you very much, Ms. Candidate, Ms. Schöittler, for your clear presentation, well illustrated. And I noticed in your last slides that you like to focus on chapter 3, 4, and 5. That's what you showed there. To start the opposition, I'd like to introduce some people here who are in the corona. First, myself, I'm Professor Steinbusch, I'm the Pro-Rector, and I'm assisted by Professor Bos, who is the secretary of this event. But much more important is, of course, the promotion team who sits opposite of me. And we have, for the audience, 3 members in the opposition, in the promotion team. We have Dr. Loosen, Dr. Manet Damas, and of course the first promoter, which will be Professor Martinez. They have guided you in the last 5 years that you today can defend your thesis. To say this, we're going to start with the chairman of the assessment committee, and I would like to give the word to Professor Rutten, who is professor in psychiatry at our university. Professor Rutten.
Thank you, Mr. Pro-Rector. Dear candidate, my big compliments to you and your work, and also to the promotional team. Reading this thick thesis, I also was impressed by the extent of the studies that you've performed. It's really translational work from molecules to cellular systems to behavior. And I also realized that you did this in a period of time from 2019, which was challenging, particularly if you would like to do experimental animal studies. So this is also, I think, a test of frustration tolerance and persistence, and that you have completed by providing this wonderful thesis to us. I read it with great interest, and I also saw your wonderful presentation. So first of all, I was struck by the cover, which you didn't explain in the booklet, or also not during the presentations. Can you, because it's autumn, also maybe explain a little bit why this cover?
Dear highly esteemed opponent, thank you for your kind words. Yes, I can of course explain the cover. For me, trees symbolize strength, and trees get their strength, of course, also through their leaves where they catch the sunlight. And for me, in myasthenia gravis, the ACHR receptor is lost, at least in the ACHR myasthenia gravis subtype. And trees lose in autumn their leaves. And for me, this kind of is a symbol for myasthenia gravis. You lose in myasthenia gravis ACHR receptor, the trees lose their leaves. And this is kind of the symbol for me. And on the back cover, it's kind of the 3 stages. You have full receptor, you have the full leaves on the trees, then you have the medium state, and after a while, if you have really intense symptoms of myasthenia, you actually have lost a lot of ACHR receptors. The tree has lost a lot of his leaves.
Yeah, you hope to provide green new leaves to the tree, right?
Of course, of course.
In the end. Okay,
I tried to implement the complement system, I tried to implement treatments, but I was not that creative anymore.
Okay, so thank you very much for this explanation. I also was struck by, so you did a lot of animal work, right?
Yes.
And in chapter 6, which you didn't present in your presentation, I was impressed by the scientific soundness of doing something that everybody would think you could just do this. No, if you're immunizing an animal, so you're hurting and you're pricking a foot pad, which you will be testing later on, and you replace it by not the foot but some other tissue of that same animal. You did this very extensively, and you also got it published, I saw. So compliments for this. But how do you see this balance between research ethics in the number of animals used, or female animals, and you also have to generate them and so forth, versus, did you need a scientific study to do this at all? I was wondering. Please, if you can comment on this.
Yes, indeed. We, of course, used animals to test a refined model. On the other hand, we hope to improve the whole model for the future, where we could save then also a lot of animals. So we had to invest some more animals to basically save more in the future. Is this answering your question?
Yeah, I was expecting this answer, so there's no wrong answer to the question. I was just wondering, how did you balance this? And I was thinking maybe you could just do the hock immunization and just say, we did it with less animal stress, and therefore we chose to do this. But you did this very nicely. So let's continue to the next point. So in chapter 3, you did this silence RNA approach, and I was again struck by the soundness of your scientific work. So I was reading that you also used a placebo off-target silencing RNA arm, and you also used 3 animals for this, and then you presented some of the work on this. But I was thinking, this is a great control group, and I was wondering, how did you develop this placebo non-target silencing RNA construct? How did you develop it? And B, did you follow up on this as a separate control group, or did you stop using this kind of an active control, right, in the rest of the experiments?
Chapter 3 is the PTMD, right? You're talking about the PTMD project?
Yes.
The acute model?
On page 78, there's the table where you list all the animals used and the treatments that you have tested. And one of the treatments was placebo off-target silencing RNA. So,
I see. You talk about the dose range finding study.
Yeah.
Yeah. This study was fully covered by our collaborators, Apellis Pharmaceuticals. We did include this in this paper because we did also include their authorships in this paper, but we did not develop the off-target siRNA ourselves here at university, unfortunately.
Oh, so you can't answer, but
do
you agree with me that this is perhaps a better control group than the PBS treatment?
Yes, of course, this is much better.
Okay.
I think, especially if you proved before that the siRNA, this placebo siRNA, does not have any effect. This, of course, needs to be tested. On the other hand, for our purposes, we only wanted to we only injected PBS in the later study because it was already tested that the siRNA was safe. We knew the toxicity was okay. We knew there was no hepatic impact on the siRNA. So with this, we could go back to a more easier control group, basically.
Yeah, it's an easier one, but it's less specific in the sense of the comparison is less specific because you have PBS versus a construct which is off target versus one that is on target. So that I think would have been an improved design. But I think it is to be complimented that Apellis already did this and tested. Then I think a last more open question. I was thinking. I can pick out of these two. I will go with this one. So you work with in vivo experimental systems. There's also in vitro experimental systems, which are currently quite developed, and you have monocellular systems and multicellular systems. So briefly, could you compare your work? Could you do some of this work already in these cellular systems that are currently developed, or you think it's too soon to do this?
To be honest, I'm not too familiar with these in vitro systems. What we tried, or what is also done in myasthenic research, is working with, for example, thymus cells, so in vitro from patients' rest material. However, these studies do never really do the translational part, really. And we really wanted to test, especially in the chronic model, the immune system, which cannot be tested in vitro so far, at least not in the models currently available for myasthenia.
Okay. So with that, thank you very much. I would like to give the word back to the prorector.
Thank you very much. Then the opposition will be continued by Dr. Denees. Dr. Denees was also a member of your assessment committee, and she is assistant professor at the Department of Psychiatry and Neuropsychology, and I'd like to give her the opportunity to discuss with you.
Thank you, Mr. Prorector. So, dear candidate, I would like to congratulate you for this great presentation and your very nice thesis. I enjoyed reading it. And I would like to congratulate your promotion team as well. But I'm here to ask you some questions. So the first one is in chapter 3, so you nicely show that your siRNA decreased the level of C3 in the plasma and in the liver. But you are looking at the disease targeting the neuromuscular junction. So why didn't you look at the neuromuscular junction, the level of C3, and would that have impacted your conclusion or the outcome of your study?
Dear esteemed opponent, thank you for the kind words. To answer your question, indeed, it would be very interesting to look for C3 at the neuromuscular junction, to look for deposition of C3 at the neuromuscular junctions. However, to be honest, the tests for this are not very sensitive or very reliable so far. We know the MAC staining, so the membrane attack staining, is very reliable, and we assume since C3 is just is just upstream of the membrane attack complex, we could use the downstream part, which is basically the cell-damaging protein. We could also use this because this is finally what we want to avoid. This is the goal to avoid in this whole treatment strategy. And for the future, yes, it would be definitely interesting also to look for C3s, but in particular, as well as other complement factors, like C4b, for example, would also be a very interesting part.
Yeah, and linked to that, because then in the discussion you mentioned that you might still have C3 at the neuromuscular junction produced by macrophages, or that you have a bypassing system that will activate C5 directly. How would you test this hypothesis in a future study design? How will you test whether these are still the case?
Thank you. That's indeed, we tested it already. Okay. So we didn't test if we had C3, but after I submitted my thesis, we moved forward because we were wondering exactly the same question. We wanted to have answers to our open questions, and we did a macrophage staining, and we could prove that there are macrophages at the neuromuscular junctions, indeed, which is very common and very. It is known that macrophages invade the acute invade the neuromuscular junction during the acute phase. We also could prove that we have indeed C3 upregulation in the muscle area, so within the muscle. We assume, therefore, that C3 comes from the macrophages. This would be the last thing to do, for example, with a double staining between macrophages and a double staining with a macrophage marker and a C3 marker, indeed, in a muscle slide to prove that it is correlating. This would be like a third step, let's say, to finally approve it, but so far we can be already quite sure that it is the macrophages that produce it.
All right, so that's not then the bypassing. You don't think
no,
that's the...
Indeed.
Okay.
Well,
it could be,
we could
adaptation?
exactly. We cannot exclude the bypassing in this moment, so for the acute model as well, but we can include the macrophages, let's say like this.
Okay, thank you. So the next question, so as you use a siRNA targeting the systemic level of C3, and that the complement system is very important also as a defense against pathogens. So how do you see using this in the future therapeutics for patients and see the balance between the treatment outcome and the risk of infection for the patients? So how do you see this as a relevant treatment in the future?
Yeah, we know that complement inhibition also comes with a higher risk of, for example, bacterial infection. However, comparing it to the current standard treatment where we use immunosuppressive treatment, which is suppressing everything basically, so really lowering down the immune response, the complement system is only one part of the immune system. It would not affect that much, for example, the purely innate immune system. The antibodies in general could still function. And additionally, there are vaccinations which are already in use. For example, one of the most common complications, bacterial meningitis, which can be vaccinated, and it is also recommended or even necessary to do this vaccination before the complement treatment is started with the current complement inhibitors which are on the market so far. So this is kind of the prevention then.
And do you see maybe using a more targeted approach with local C3 inhibition in the muscle and not systemically?
Yes, indeed. For example, inhibiting the receptors specifically on the local tissue which is affected, and here in this case it would be the muscles, would be, I think, a very ideal target, because this on the one hand allows the specificity of the target, and on the other hand, allows the rest of the complement being active.
Thank you. Do I have time for another question?
Yes, you have much more time.
Okay, thank you. Then in chapter 5, you emphasize the importance of using the right animal model to target your research question. So here in your thesis, you use several models that showed different outcome with your siRNA for C3. So I would like that you elaborate on it for you in the future, which would be the ideal model that you use for myasthenia gravis research, and especially analyzing the C3 or, in general, complement system, based on the different outcome from the model that you had.
Yeah, so for myasthenia, I still think that both models are very important, because the model characteristics are just different and depend on the research question. You might need one or the other. To compare a bit the models a bit more in depth, the acute model is a very reliable model with lost a little variability between the animals usually. So for starting studies, so for example, testing if a proof of concept basically, if a treatment at all would reduce or would have an effect in myasthenia, I think this model is important still. Furthermore, it really represents the acute phase and the myasthenic crisis, which is also in patients a problem. And for this, I think this model is still very, very good to use. For the long-term effects and for, of course, how the immune system actually defends against myasthenia or what actually happens, the processes, the chronic model is indeed the more the one to choose, because you have the whole process. You have you immunize with the antigen. Basically, the animals have to develop their own immune response. This part is totally missing in the acute model. And with this, you also have the complement activation and very in-depth, no, how to say it. You have basically every part of the immune system active, which is comparable to the situation in patients. And I think the acute model is never enough. You always need the chronic model, for example. Vice versa, if you're sure that your treatment is working and that it has an effect on patients, you could, for example, also move directly to the chronic model. Did I cover your question?
Yeah, but also do you think maybe that at the end you might have different treatment targeting the acute versus chronic, because the mechanisms are different?
Yes, indeed. So I think, for example, C3, as I said also in my summary, C3, I personally, based on these results, are more for the acute phases, acute crises, for example, in myasthenia. We didn't see a long-term effect. We saw some trends, but we didn't receive too many significances, and we saw, for example, muscle strength was not affected. So I think in a long-term effect, this treatment against C3 would not be ideal. And therefore, yes, depending on the phase you want to prevent or to treat in patients, also the disease would change. The treatment.
Okay, thank you very much. I'm satisfied with the answer to the question. I give the word back to the prorector.
Thank you very much. The opposition will now continue by Professor Frausen. I hope I pronounced it correctly. She is professor in the Department of Immunology and Infection at the Biomedical Research Institute in Hasselt. And I, of course, like to thank her that she came all the way from Belgium to the Netherlands. And I'd like to give her the word.
Thank you. Dear candidates, I would also like to start by congratulating you and also the team of promoter and co-promoters with a very elegant and well-written PhD thesis. I think the work presented in the thesis and here today is clinically relevant. It has direct translational potential and was very well performed, so congratulations for that. And I would like to discuss some aspects with you from chapter 3 that were already touched upon by the previous opponent and that you also presented today. Namely the fact that the C3 targeting could not completely intervene in the loss of or could not completely block MAC deposition at the neuromuscular junction. And indeed, one of the theories that you proposed was that other cells locally might still express C3, and you just mentioned that you already showed that macrophages were present. So my question is, have you already thought about potential strategies of how you could extend your targeting approach to not only really target hepatocytes, but also act more locally on cells at the neuromuscular junction, such as, for example, macrophages? Can you think of strategies to do this?
Dear highly esteemed opponent, thank you for the kind words. If I understand your question correctly, you asked for improved or advanced strategies to target not only the C3 in blood, in liver basically, which is soluble then afterwards in blood, but also local C3 production like in macrophages.
Yes.
Is this correct? Okay.
Yeah.
I think, so for at first with our treatment, of course, with this siRNA construct, we cannot do this, but thinking further, there might be, for example, options to link an siRNA. It is also done in other studies where an siRNA is linked to other compounds which target an additional receptor, for example. I could just think about that, for example, if you want to target, for example, the C3 receptor in the muscles or in the macrophages, specifically, depending on what you're looking for, that you could link it to a specific marker, for example, then to the macrophages as well. So it basically has to bind both targets to become active or to actually work at this position.
Do you have any suggestions for potential receptors?
The macrophage, what was it, 60? There is one, which I don't have in mind, 65 for macrophages. CD6, I must say right now, I don't have it on top of my head.
It's okay. And then the other potential explanation that you had is that there might be a bypassing mechanism for complement production. Could you target that? Would you continue with C3 targeting, or do you think that's maybe not the best complement factor to then target, or?
Yeah, indeed, there are bypassing mechanisms, especially in the last years during our studies, indeed. Also some other publications showed that either C3 inhibition but also C5 inhibition did not fully prevent the membrane attack complex at the junctions. And everyone is basically thinking the same. There must be other mechanisms avoiding these targets. And this bypassing mechanism is so if you inhibit C3 or C5, both of them actually, it turns out that C4b, which is a convertase of C3, so you have, I don't know how familiar you are with the complement cascade.
I'm familiar.
But the protein which basically cleaves C3, part of it is C4b, and if this accumulates and if the level of C4b increases, it was shown that it can directly activate the terminal pathway. And with this, it would, of course, become one of the very prominent targets to inhibit, or even do a double inhibition, not only one of the complement factors, but, for example, two, because it seems to be that there's some redundance. If the complement pathway is inhibited at one point, that another so-called bypassing pathway, I think we rather have to call it in the future maybe, the pathway, that those become more active and more prominent. And with this, we would, of course, need a target or two targets which target each of these options. And then, for example, C4b, with the current knowledge, would be one interesting target to do. Yeah.
Okay, thank you. I have one small question still related to this. So the rationale also for targeting C3 instead of, for example, C5 is to also avoid the production of anaphylatoxins and like pro-inflammatory cytokines. Did you check production of pro-inflammatory cytokines in your models? And if no, how would you do that? And which cytokines would you look at and why?
Short answer, no, we did not check for this. We know there are anaphylatoxins which are released, for example, C3A. However, testing, for example, you can test it in blood and serum and plasma with ELISAs. The assays are available. And I think this is indeed something very interesting to check if these are really relevant, if this is a relevant aspect to consider during the inhibition. Did I answer your question?
Which cytokines would you then check? Which ones are relevant in the pathogenesis?
yeah, C3A, for example, is one of the anaphylatoxins, C5A as well. These are, at least based on literature, the most important ones, which have pro-inflammatory effects.
And if we really look at cytokines?
Cytokines, sorry.
Which ones would you?
IFN gamma is probably one which might be interesting or important to check. IL-6, I would also consider. And on top of this, I must admit I would go into research, literature. I don't know on top of my head.
Okay, and then I have one last more general question, because your thesis really focuses on complement and autoantibodies in the pathogenesis of myasthenia gravis. This is all linked to B cells, but B cells are known to have other functions as well, except their involvement in antibody production. So we call these antibody-independent functions. Can you tell me something more about these functions and whether they are also relevant in this pathogenesis or not?
So functions of B cells that are not the antibody production.
Yes.
This right now is out of my expertise.
So they can, for example, stimulate T-cell responses by antigen presentation and providing co-stimulatory signals.
Okay,
Is that also relevant in the pathogenesis, for example?
Of course, sorry, yeah. These, of course, I thought something totally different. So the interaction with T cells might also be, could be a relevant factor in myasthenia gravis, because we know the T cells are developed in the thymus, where they are programmed wrongly, and that if they then interact with B cells, the B cells are activated. So to interfere with this function, this would be also helpful, I think, to prevent the autoantibody production in the next step then.
Okay, thank you. I would like to give the word back to the prorector.
Thank you very much. Then the opposition will be continued by Professor Bos. Professor Bos is the chair of the Department of Internal. He works in the Department of Internal Medicine, but he's the chair of immunotherapy of cancer at our hospital, and I'd like to give him the opportunity to discuss with you.
Thank you very much, Prorector. I was very happy to receive a thesis on myasthenia gravis, because as you know, there is a long history on myasthenia gravis in the laboratory of immunology where I still work. That was started with Professor van Breda-Vriesman, who unfortunately passed away a couple of weeks ago. So I think we should remember him here first. And he was from Leiden, which is completely different than from Maastricht. So we would never compliment a commission and the candidate before she has proven to defend it accurately. But I'm happy to make an exception with that move. I was intrigued by page 41 because as a hematologist, I worked on graft-versus-host disease, bone marrow transplantation, and you mention here that bone marrow transplantation might indeed resemble autoimmune disease. And at that time, I did transplantations in the lab, and Jan Verschuren, who's now the professor you probably know well in Leiden, on myasthenia gravis and other neurological diseases, he worked on myasthenia, and I checked all his animals. And certainly in my case, there was an animal after a bone marrow transplantation that had myasthenia gravis. And we published that, and there is no thymoma in this mouse model. And you refer here to a thymoma-associated multi-organ autoimmunity. I think that's not the correct term. We had another term. We published that 4 years before you were born. So I don't blame you that you missed that publication. Okay, let's go to the serious stuff. Well, this is even serious. Okay. Everybody talks about THREPTA 3 and 4. I would like to do that as well, but from a methodological point of view. Because from immunology, especially in cancer, we all know that everything works if you do it preventive. And nothing works if you already have the disease. So my methodological question is, why at all did you start with chapter 3? Because you will know it works.
Dear highly esteemed opponent, thank you for these kind words. From my perspective, I must say, as a new PhD candidate, I did not know that everything preventive would work. I think, more understand that it could work, because if you prevent already the mechanisms that lead to the disease, you have it much easier, of course, to not see any symptoms than when you have the disease already developed and then you have to kind of fight against it. However, we were not only interested in this project to understand or to see if the treatment worked, but we were also interested in the effects which it would have on a myasthenic level, so more in the effects of myasthenia, while the company, of course, had much more interest in the treatment research. And we wanted to investigate actually what is complement doing if we inhibit one of these factors, what is happening with myasthenia then. And also, initially, maybe it's too far now, but initially there was also the idea to find maybe some biomarkers to find changes which happen. This didn't happen, it didn't end up in the thesis or anywhere, but since you asked for the idea, why we had this idea, I think this is important to mention, that we wanted to see if we can predict also myasthenia maybe. And the acute model especially is resembles quite well the acute myasthenic crisis phase. And with this, we hope to get some insight into this direction, if we could also predict this.
And then you switch to chapter 4, after you discussed in chapter 4 that these models are completely different. So why do you switch to another model if you want to see whether it's not only preventive but also in the real phase of the disease? Why wouldn't you stick to the same model?
You mean why we didn't stick to the acute model, the PTMG? Yeah. But well, in the PTMG, we indeed only we inject an antibody which blocks the receptors. With this, we do not represent at all the immunological processes that happens actually in myasthenia. And the first one was more or less a proof of concept that if we inhibited complement, that it would have an effect, it would have an beneficial effect for the myasthenic rats in this moment. And then, but we were of course interested if we could also have the effect in the actual disease, if we only inhibit it afterwards, after the disease starts.
You see that your first model, where you state that it's also the crisis, it resembles the crisis, is not a real model for a crisis?
Sorry, again, can you repeat the question, please?
If then is your first model, because you conclude that that might be an accurate model for a crisis, might not be an accurate model for a crisis then? Because the patient comes in with the crisis and you don't know 2 days before. So why didn't you wait until you had the crisis in the animals to start the treatment?
I think this is, for me to answer, I have to answer basically with two things. The first,
you can make 3 things. I don't care.
The first thing, as I said, the acute model, I think it represents indeed the crisis from the pathological mechanisms which happen there. We can, of course, not predict, not in patients, predict when the crisis happens. However, we can see the macrophage invasion, for example, and this one we also have in the acute model, and we could see, or we could test if the treatment would, for example, prevent it, if it would have an effect on this macrophage invasion. And for this, I think it is still a very good model, experimental model. For a patient-oriented translational work, of course, the chronic model is the one we have to go for as treatment, since there we only administer the treatment after the disease starts. So even if a crisis happens and we only can treat it, if a crisis happens in the patient, we can only treat it afterwards. And this is what we tried with the chronic model, basically.
Can you ask one of your paranyms to read proposition 1 for me?
The rapid effect of C3 inhibition offers a promising strategy for managing myasthenia gravis crises effectively.
So after what you explained to me and the other people, would you still, and I know the rector wants me to ask a different question, would you still take this as number 1 proposition after everything you explained to us on the relevance of this method when the patient comes in with a crisis and do not get the preventive treatment? Do you have evidence in your thesis for that?
I think based on the rapid effect, which we saw the treatment being effective, so C3 was downregulated within 24 hours after administration, the mechanism is still, yes, for the mechanism, yes.
But but the house is already burning. You explained it quite nicely. The damage is already there.
Yes, but if you inhibit then C3 in a crisis, so C3 would not add any further damage on top, right?
Okay, you see, I was trained by Professor van Breda-Friesman, and he was not a nice guy when he had these kind of positions. I'm going back because I see that you really like mice and you don't want to kill them.
Sorry, I like what?
You are rats, you like the animals and you like to prevent their damage and whatever harm. Why on earth should we go with mouse models? And if we do, because there are so many patients, if we go to patients, what would be your idea to start off with? Because it's about B cells, it's about antibodies, it's maybe complement, but that's the end of the cascade as far as I know the immunology. What would be your idea to do a new clinical trial on patients?
If I understand your question correctly, first question was why we would use mouse models?
Skip the mice.
Okay.
What would you like to do in patients?
You mean as a treatment? Which treatment I would apply?
You got a new grant, and you go to Germany, they give you 250,000 euros. What would you do?
I think I would, with the current treatment, sorry, I don't understand your question,
With your brains and your ideas. What is your ideas?
Okay.
How to spend your 250,000 euros?
I think I would go indeed into double suppression, not of C3, but more into, for example, which I mentioned before, C4 or C5, which is already known to work also in patients.
We do start a trial in Maastricht with autoimmune diseases like lupus and things like that with a CAR NK cell.
Mm-hmm.
Do you expect that the clinician will participate to that trial? And what CAR would you like to use for that? You know what a CAR is, huh? A chimeric antigen receptor NK cell.
Yeah.
We make them in our labs, so you can
Yes.
test them there. Would you advise the clinician to participate into that trial if you have the money?
I think yes, because CAR NK cells are indeed also very, it's a new approach, but I think quite promising. We know NK cells have an impact in myasthenia, even though they are not very well discovered so far. We know there are some upregulations for some specific markers, some downregulations, but I think it is an interesting and also promising.
But the NK cell doesn't have to be part of the pathophysiology. You just make them in the lab and give them to the patient, the CAR NK cell, as a treatment.
But it would be nicer even if they would have a double effect, right? If they themselves, no?
No. Okay, that's for the drinks. I'll come back to you. I would like to give the word back to the pro-regular. Thank you very much.
Thank you very much. And then we continue to the last opponent, which will be Dr. Huy Marcus. She is involved in the Department of Neurology, and I'd like to give her the opportunity.
Thank you. Dear candidates, I also want to like to congratulate you on this wonderful thesis and also the congratulations for your supervising team. And as a clinician who regularly treats patients with myasthenia gravis, I'm very pleased with your thesis as it represents another step towards improved treatment, and that's great. And given my clinical background, I personally have no experience conducting animal studies. And regarding the animal studies, Proposition 10 caught my attention. So I would like to ask Paranenf to read Proposition 10.
Some people talk to animals, and not many listen, though, and that's the problem.
Yes, I talk a lot with my patients, and I know how important it is to really listen to them, and I think I do that. And to assess the efficacy of treatment, patient-reported outcome measures are therefore increasingly used instead of only using the muscle strength. And nowadays in clinical trials with myasthenia gravis, we use a lot the MG-ADL. And in chapter 3 and 4, you use muscle strength measurements and also a disease scoring. But what I miss there is the speech. So yeah, what's there to listen to, because you don't look at the speech or you don't listen to the animal, as well as the ocular symptoms like double vision and ptosis. You don't check that in your animals. And well, those complaints in patients, they really impair the functioning. So what would you propose to improve this animal model to also capture those same symptoms?
The esteemed opponent, thank you for the kind words and your question. Addressing also the proposition, because you also addressed this, I think it is important to listen to animals, but in contrast to human, we cannot ask them questions. We can only react on their actual behavior. And this is also what we had include in both of the studies. We checked the welfare, we checked the disease scores, which include welfare assessment. So we checked for general disease signs, we checked for walking behavior, for example. You asked for the ocular things. It is quite difficult to ask an animal if it has double vision, of course. So this is a struggle, I think, in animal research, that we cannot just copy the tests from human to animals, of course, and therefore we have to, I think even more important is therefore to listen to the animals, to observe them, and to actually react based on their reaction. So if we see an animal is sick, it will, for example, try to cover it. Especially mice will try to cover it. Rats show it more obvious. And then it is on us to handle sufficiently and to handle appropriate. And you asked which tests I could further include. Is this right?
Yeah, clinical tests in the mice.
Yes. So I think strength test, at least in the acute model, there we have again the difference. The acute model, I think we saw very, very impressive results. For the chronic model, it is not always the ideal test, let's say. Also, we didn't see very, very huge impact on the rats. We also included the EMG in the electromyography in the end. So this we could, for example, also do midterm. Not with curare, so not with the inhibitor then included, because then we can't wake the animal up anymore. But we could include this one or maybe even 2 times during the experiment to see, is the neuromuscular transmission already impaired? At which point is it impaired?
But then with the EMG, you also check the same muscle, so it's the same as the muscle strength. Can you also do an EMG with the eye muscles?
Not
in patients, we do a single fiber EMG. I don't know if it's possible in rats, but
if in mice, you can do that. To be honest, I don't know if this is done in animals, if the ocular nerves or ocular muscles are affected. However, for the EMG and the strength test, that are different muscles. We do the strength test in the front pores and the EMG in the hind paw. So this is already 2 different muscles which are affected then.
Yes.
For mice, we have more tests. Also, we could, for example, the inverted mesh it's called, so they have to hang upside down on a mesh. For the rats, we also test their climbing ability, which is not a one-time occurrence, but they really have to climb up on the hand, affecting basically the whole body, because if you climb up a rock, you need every muscle you have. And with these tests, we indeed can already test for strength loss or for weakness, much more sensitive, of course, in combination of all of them.
Yeah. So you don't hear they make different noises after treatment. Like the patients have a dysarthria, they have speech problems. You don't hear a difference in your animals.
We see and hear different noises, and also in the they breathe. Their breath is sometimes different. Their skin is different, so we see general disease signs. We see the weakness actually on the body composition of the animal. They are totally weak and very, very.
Then
you don't want to grab them actually, and these things we see. We also see in their eyes. It's a general sign of weakness, a general sign of the disease. Sorry, we see red eyes, for example. This tears basically getting red, so we see very clear symptoms. However, the eyes, for example, are not specific to myasthenia. This is to mention. That's
but it's good to hear that you look broader than only the muscle strength.
Yes.
And another question about the animals. You already talked about chapter 6. There you demonstrated that hoc immunization in the mouse EAMG model leads to less discomfort for the mouse. And on page 193, you state that using mice offers advantages over using rats. But why then did you choose to use the rat model in chapter 4?
This question is very easy to answer indeed. The previous testing which were done by our collaborator were already also done in rat. And for the first test we decided, this was one of the reasons, to continue in rats, to not change also the model, to not adapt too many different things, because then we cannot compare it, of course, to the outcomes from before. And also the whole toxicity testing, all this has to be done in these rats. So.
Okay, but for a new study you would prefer to use mice? Or not?
Sorry again?
If you want to do a new study, do you want to choose for mice or rats for this model?
Personally, rats, they bite less. No, I think actually both would be nice to see. On the other hand, complement-related research is better in rats indeed. So I would continue with some rats, not only personally.
Okay, well done. Yeah. Then the question is what you want to do with the results of chapter 6. Yeah. Then at page 118, you mentioned that the current complement inhibitors requires frequently administration and that they both lead to high healthcare costs. But I was wondering, what would be your recommendation regarding the frequency and the mode of administering C3 inhibition?
So what is known from siRNAs, for example?
Dear Ms. Candidate, you are allowed to answer the question, but you also have the possibility to stop here. It's your choice. Would you like to answer this or stop?
I can shortly answer, yes. So siRNAs, for example, what is known so far, they have to inject it less frequent. In the moment, the current trials, which are ongoing in patients as well, there's injection, I think the highest frequency is every 3 weeks, but they are going up to 3 monthly, 6 months. So, and with this, we would, of course, also highly increase their life quality for the patients if they don't have to administer it on a daily basis, for example.
Okay, thank you for your answers.
Thank you very much. Dear Ms. Candidate, dear Ms. Schöittler, the time appointed for defending your thesis has now officially passed. The degree committee will now withdraw to discuss not so much the quality of your thesis that has been approved, but we discuss the way you have defended your thesis, but we also will discuss the quality of your presentation today. I request that you and your company await the results of our deliberations and the return to the aula. Thank you very much.
hormones.
They're so healthy, so German.
Yeah, I'm hungry.
You can have both of us.
Yeah.
Dear Ms. Schöittler, the degree committee here present has assessed the quality of your thesis, but in particular the way you have defended your thesis this morning, and also taken into consideration the presentation you have added to your defense. In view of its positive verdict and taken into account your previous qualification, the degree committee has decided to grant you officially the degree of doctor. And see your smile on your face. The body language is pretty clear about this. The Professor Martinez is authorized to confer upon you this academic distinction in accordance with Dutch University customs, and therefore invite your supervisor now to take the floor and to do the necessary obligations.
Thank you. So, do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Anja Kristina Schöittler, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with a degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university.
Can you please turn it?
Yes.
For the picture. The laudatio will be presented by Professor Martinez.
So, allow me to begin with the words of the great German scientist Paul Ehrlich, a Nobel Prize in Physiology of Medicine in 1908, and one of the founding fathers of modern immunology. Ehrlich, who pioneered the concept of the magic bullet in medicine and laid the groundwork for our understanding of antibodies, once said in German, but I'm not going to read it in German, I will translate it in English. In science, the attempt must be made to reach the goal, not because it is certain that it will be reached, but because it is worth the effort. So these words perfectly capture the spirit of scientific dedication, and they resonate deeply with you when we speak today about Anja. Anja, a remarkable student, originally from Ratingen in Germany, has from early on combined academic curiosity with a strong sense of civic responsibility. During her voluntary work with Anfallhilfe, she gained valuable experience in medical transport and emergency services. This commitment not only reflects her dedication to helping others, but also provides her with profound insights into the importance of teamwork, resilience, and care in critical situations. Her academic journey has been characterized by a broad interdisciplinary interest. She explored diverse fields ranging from ecology to neuroscience, never limiting herself to a single perspective. This openness and intellectual versatility has allowed her to develop a truly general understanding of science and its societal impact. Her passion for research, particularly involving animal models, began at a very young age back home in Germany. She and her family once discovered mice in their garage. So sometimes this is not nice, of course, but with curiosity and creativity, they decided to label the mice and released them several kilometers away, only to find out that the mice returned home surprisingly quickly. So this simple but insightful experiment planted the seeds of Anja's lifelong dedication to science and her fascination with animal behavior. As Konrad Lorenz, one of the founding fathers of modern ethology, once said, every animal leaves traces of what it was. Man alone leaves traces of what he created. This early experiment saw Anja that even the smallest of creatures can reveal great truths about behavior, learning, and adaptation. As Anja further broadened her horizons at our department, but also before that, she was in Cambridge, where she learned and immersed herself in cutting-edge research, and it was very well enjoyed there. I learned. So late during her time at Maastricht University, she not only endured the challenges of a global pandemic, but also carried out many demanding animal experiments, which are beautifully illustrated in her PhD thesis. This work required perseverance, resilience, and not least a strong sense of discipline. Quiet and always deeply concentrated, Anja is also highly perfectionistic. Every experiment had to be organized and analyzed with the utmost precision. Nothing was left to chance, and her careful approach ensured that her work stood on a solid and reliable foundation. Of course, Anja is not only a scientist, but also a true ambassador of her culture. As a good German, she enjoys frikadelle and other culinary delicacies from her homeland. With generosity and humor, she even taught us online how to prepare some of these dishes, leaving us not only scientifically enriched, but also gastronomically delighted. And when she is not in the lab or the kitchen, she finds her balance in swimming, a sport that perfectly reflects her energy, endurance, and determination. What stands out most in Anja's trajectory is not only her academic excellence, but also her ability to rise stronger after every challenge. Anja has shown that setbacks are only the beginning of new strength, and that perseverance is at the heart of her scientific journey. And yet, at the end of the day, we scientists do not do this work to be loved or to be popular. Very often we will be misunderstood, unappreciated, or even criticized. What truly matters and what is truly admirable is the dedication, the determination, the love, and the passion for doing good and relevant work. And this is what science is about, and what I wish for you, that you continue to carry these values forward wherever you might go in the future. Congratulations, and also to your family.
Thank you.
So, dear Dr. Schöittler, I would like you use now your official new affiliation. I would also like to congratulate you on behalf of the board of directors of our university. In my congratulations, of course, also your promotion team who guided you, that you could shine today, and you did. I would like also to thank the members of the Corona who addressed questions to you. And of course, they only had 9 minutes, but it takes more than 9 minutes to prepare these questions. And I think I would like to join them. And I also like, of course, particularly thank Professor Frausen that she came all the way from Hasselt. In my congratulations also go to your family, also schöne Grüße an die Familie hier, die aus Deutschland gekommen ist. Wir sehen uns später in der Refter. I would like to also mention something about myself personally, which I read your thesis, if I may. And I was a little bit surprised. We can see here on the screen the 3 trees which you have. So you have the trees going from, I would say, from autumn into winter. Yes, not from death to not from alive to death, but in winter. And I would expect it, and maybe you don't have to answer this one. It's not a question. But if you go to chapter 9 and 10, which is the impact factor, and your personal CV, you put your personal CV with a completely winter tree, which is a dead tree. And I would have expected that there would be a tree with some slowly coming green leaves. So there is new life coming up. You did this and you're going to continue. So we discussed this later at the rafter, but I was surprised that you didn't put yourself much more alive than in your thesis. Anyway, saying this, we're going to conclude. And how do we proceed? We're going to we proceed as follows. We as Corona, and of course with the new doctor, will make a picture on the stairs of our university. And we will join you later in the rafter. So you can already go to the rafter, enjoy some coffee, tea, maybe some pie. I have no idea what she has ordered. And we will join you in approximately 5 minutes. Thank you very much. And thereby, I officially close the ceremony. Okay.
De Duitsers nog geen biertje voor zetten.